We're now in the third part of our Rickettsia, Coxiella, and Bartonella lecture. And in this lecture, we're going to focus on the Anaplasmataceae family. Now remember, the Anaplasmataceae family is a different family than the Rickettsiaceae family, although the organisms do have some similarities. So one of the genera within the Anaplasmataceae family is the genus Ehrlichia. And Ehrlichia canis is the etiologic agent for the disease ehrlichiosis. Now, the thing about Ehrlichia is it has a developmental cycle that's very similar to that of chlamydia. So there is an infective form of the organism that's called an elementary body. And there's an intracellular form that's called the initial body. So it's really important with these organisms that have developmental cycles that you know which form is the infective form and which one is the intracellular form. Now, the vector that transmits Ehrlichia canis is the dog tick, and the infection resembles Rocky Mountain spotted fever. So the symptoms include a fever, muscle aches, there is a rash, not eating, your typical type of flu-like symptoms. Now, we already said developmental cycle. The Ehrlichia is very similar to chlamydia. You have an elementary body. That is the stage of the Ehrlichia organism that will infect individuals. What happens is then the elementary gets into the body, gets inside cells, and then what happens is it generates inclusions. So then it changes its stage and it develops into what's called moruli. So what happens is these inclusions inside the white blood cells get very large. The host cell ends up rupturing, and these moruli break apart into a bunch of elementary bodies, and then those elementary bodies continue that cycle. They go on and infect nearby cells. They end up developing into moruli. The host cell ruptures, the moruli break apart, and more elementary bodies go on to infect more cells. So here are white blood cells, and you could see in the cytoplasm these inclusions, and these are the Ehrlichia organisms within the cytoplasm. So these are very difficult to identify in the clinical laboratory because white blood cells can get inclusions. There's various diseases that generate different types of inclusions. Ehrlichia commonly can look like a platelet just sitting on top of the white blood cell. So unless someone is infected with a large number of Ehrlichia, it's very difficult to find these organisms in white blood cells. Here is the morula in a white blood cell, so they get very large and just start to spread through the white blood cell until the cell bursts. Again, a nice large inclusion in the cytoplasm. The other organism in the Anaplasmataceae family is Anaplasma phagocytophilum, and this causes a disease that's very similar to ehrlichiosis. There are approximately 16 cases per 100,000 in the population in northern Wisconsin between 1990 and 1995. So it's not a very common infection, but it is more common in certain regions of the United States. So it's more common in the upper Midwest, such as northern Wisconsin, northern Minnesota, as well as the northeastern region of the United States. Ticks are the vector of this disease, and specific ticks, Ixodes scapularis and Ixodes pacificus, are the vectors that transmit anaplasma. Now, the natural hosts are deer, rodents, horses, cattle, and humans can be a host. So what happens is in areas where you have a lot of deer, like the upper Midwest, northeastern US, where you have a lot of wooded areas, you have a lot of deer, you have a lot of rodents, so you also have a lot of ticks. And if any of those get infected with anaplasma, you then get humans that are going to, go on nature walks, go hiking, things like that, and pick up infected ticks, and that's how the cycle continues. So our next organism that we're going to talk about, and this is not in the Rickettsia or the Anaplasmataceae family. It used to be part of the Rickettsia family, but it is not anymore. And this is the organism Coxiella. And there's only one species that's important, and that's Coxiella burnetii. So this again is a gram-negative coccobacillus tiny rod, and again this is another one of our obligately intracellular organisms. So this is not going to be an organism that you are going to grow in any type of agar media in the microlaboratory. This organism also has a very complex developmental cycle where you have an infective stage and an intracellular stage, very similar to your Ehrlichia. However, the infective stage of Coxiella burnetii is called the small cell variant. This stage is very similar to a spore that allows the organism to survive in harsher environments. You also have your intracellular stage, which is called the large cell variant, and this is the stage of the organism that develops inside of host macrophages. Now, Coxiella burnetii causes the disease Q fever, and Q fever is primarily a disease of livestock. So animals are usually asymptomatic, but animals can be infected with Coxiella burnetii, and these include cattle, sheep, goats, rodents, and marsupials. So what happens is individuals get infected with Coxiella burnetii through either direct contact with an infected animal or inhalation or ingestion of infected animal products. So this is usually an occupational disease. People that on a daily basis are working with animals or animal products. Now, 50% of infections are symptomatic. So you have asymptomatic and symptomatic cases. And Q fever causes an influenza-like illness where you can get a systemic infection either with or without pneumonia, and normally the symptoms are fever, chills, a dry hacking cough, and there is no rash with Q fever. The mortality rate of Coxiella burnetii infection is 2%, and there is a chronic form of the disease. So the chronic form of Q fever is much more common in people that have had heart valve damage or people with cancer that are undergoing immunosuppressive chemotherapy, and the mortality rate of the chronic Q fever can be up to 65%. And because Q fever is associated with a high mortality rate, it is considered a potential biothreat agent. So for those of you that are making your charts of, similar things throughout the semester, you should be making a table, and in your table, potential biothreat agents, and Coxiella burnetii is one of them. There are several organisms we've talked about thus far that can be potential biothreat agents: Francisella tularensis, Burkholderia mallei and Burkholderia pseudomallei, Brucella species, Bacillus anthracis. So there's numerous organisms we've already discussed throughout the semester already that are categorized by the government as being potential biothreat agents. So you should be pulling all of these things together as a whole in microbiology. Now, Coxiella burnetii is highly infectious, and the infectious dose can be as low as one organism, again, making it a candidate for a biothreat agent. Now, if an individual were suspected of having Q fever, the lab should be notified, because you'd want to handle that specimen much more carefully. I mean, you should always handle every specimen carefully, but if it might be suspected that a specimen has something highly contagious, you really want to be careful. So remember, this organism doesn't grow in your broth or your agar media. It has to grow in cell culture. So if you were to grow it in cell culture, you can see those cytoplasmic inclusions. However, that's not commonly done in the clinical laboratory. You're normally going to diagnose these obligately intracellular bacteria and viruses using serological testing. So for Q fever, you'd have antibodies to both phase 1 and phase 2 antigens. And an individual that has acute Q fever would have a high titer of phase 2 antigens. And an individual with chronic Q fever would have a high titer of phase 1 antigens. Here's an electron micrograph of Coxiella burnetii, and you can see little teeny tiny rods. So Q fever, individuals are commonly infected by breathing in aerosols from infected sheep, cattle, or goats. So what happens is be that may work on farms that have infected animals. These animals are urinating, defecating, their milk can be contaminated with Coxiella. So if you happen to be sweeping up an area where animal excrement has been and there's dust that comes into the air, you can breathe that dust in and then get infected that way. The organism spreads through the bloodstream, and diseases that usually will occur are pneumonitis, endocarditis, granulomas can form in the organs, and this can turn into a fatal infection. So here's a little poem about Q fever. A worker from cattle plant D earned money by mopping up P. One day he fell ill with horrible chills and a fever of 123. So we're going to move on to the last part of this lecture.